Hey Beaks, it's Mr. Boylan, and today I want to talk to you about prokaryotic gene regulation. Okay, and gene regulation is amazing. Okay, this is just one of the coolest things that you could ever learn about in biology. There is so much that goes into gene regulation. Now, we're eukaryotes, and that is so cool and complex. So we'll get into that later. But today, what we're going to talk about is also really cool and pretty complex, and that's prokaryotic gene regulation. We're going to get into the Operon model. And if you think about it, gene regulation is one of the coolest things that we could possibly talk about because that's the whole point, right? Gene expression. That's what biology is all about. That's what you have that DNA in there for, is to give you all of these traits, right? And make sure they're working correctly. So this is really cool stuff today. Now, I will say, if you don't understand transcription and translation very well, if you don't understand gene expression, today won't make a lot of sense to you. So you should probably go back and master that stuff first, okay? Or watch those videos. But if you're feeling good, here we go with prokaryotic gene regulation. Okay, so here we go, conducting the genetic orchestra, right? Prokaryotes and eukaryotes, they alter their gene expression, okay, in response to changes in the environment. So think about if you just had a milkshake from Chick-fil-A. Your intestinal cells, right, your stomach and your intestines and your digestive system, all of a sudden they got triggered. Say, what is this delicious vanilla milkshake? You know, what are all these lactose sugars and things like that? Whereas even the prokaryotes that live there, you just change their environment as well, right? And now they're saying, what is this sugar? And you're going to turn genes on and off and regulate in different ways because of that. Now, prokaryotes do it very coolly and very easily at what's called an operon. But regardless of what type of cell you're talking about, even the complex eukaryotes, it's kind of like an orchestra, right? So what's going on here is you don't have somebody leading the orchestra, right? There's no one in there conducting. And that's important. Now you might say to yourself, what about DNA, right? DNA is really just a storage system, right? For genes, RNA is really the guy who's out there doing work, but there's not one RNA, right? There are millions of RNAs and many, many different types of RNAs doing their job. So gene expression is responsible for lots of different things, and a lot of the guy who does the work is RNA, don't forget. Okay? Imagine back when the sperm and the egg came together to form the zygote during fertilization, right? How did you become who you are? Gene expression, right? How did you make brain and bone and blood and eyes and all of that? That's gene expression. Okay? So what we're going to talk about today is, well, how is this all regulated? And don't forget, this took billions of years to get it as good as we are right now. We've all been evolving, right? Us and our common ancestors since the beginning of life on this planet. And we're pretty good at this. And eukaryotes have it fine-tuned at many different places. Prokaryotes, you'll see the Operon model in a second, and they can do some good too with their gene regulation. So let's take a look here. So bacteria often respond to environmental change by regulating transcription. Right now, as you know, transcription is when you make a piece of RNA from DNA. So we're talking about messenger RNA here. Natural selection would favor bacteria that basically don't waste their time and resources. If you're a bacterial cell and you're making things that you don't need, that's not good. And you know, you'll get weeded out because there's other bacteria around you that probably aren't wasting their time or resources like that. They're going to live longer and reproduce more, right? So that's evolution. You're going to get weeded out of the environment, right, of that population. So take a look what's happening here. A cell can regulate the production of enzymes by feedback inhibition or gene regulation. Okay, so what does that mean? Well, let's take a look here. We have a precursor molecule here, okay, and we know bacteria need tryptophan. Okay, remember that's one of our amino acids. And that's an old wives' tale. People used to think that that's what made you sleepy right around Thanksgiving, or if you eat too much turkey. And now we know, no, it's just because we're pigs and we eat too much. But point is, they do need tryptophan. Okay, so how do we make tryptophan? Well, you need this precursor. So somebody either had to give you this, it had to be in the environment already. Okay, now to make this precursor into this guy right here, this intermediate, okay, you had to have these two genes turned on and activated to make enzyme 1. Sure. Now to get this intermediate to become this intermediate, well, this gene hopefully was on to make enzyme 2. And as long as you have this intermediate right here, you can make tryptophan from it. As long as genes B and A are on, they'll make enzyme 3. Okay. So one way that you can stop the production of tryptophan would be to regulate the expression of one of these enzymes, right? imagine if you blocked one of these earlier enzymes. Okay, well, there you go then. And I said block there. Maybe I should have said, imagine if you regulated the expression of the DNA right here so that this enzyme's creation is blocked. Okay, so you don't even get a chance to use this DNA right here because it's turned off. Now, what about on this side over here? This is cool. Instead of regulating the enzyme production, let's regulate the enzyme activity. And do you remember what this is called? We're going to bring up the tryptophan when we have enough of it, and we're going to go ahead and block the enzyme, right? Is that ringing a bell? Remember, that could be something like inhibition, competitive or non-competitive or allosteric inhibition. You can block the enzyme, and what you just did, think about it, you shut down the metabolic pathway. Here's a pathway that tells you how to make tryptophan, and when you have enough tryptophan, what a beautiful system when the tryptophan could go and inhibit one of the first enzymes that makes tryptophan. That bacterial cell is kind of a genius, right? What a way to save resources. That's negative feedback. He's stopping the pathway by using the product, which is going to be what the pathway is all about because he already has enough of that product. Huh. Pretty cool, right? So how do they do this? Well, let's take a look at the Operon model. Okay. So the basic concept of an Operon. You have a cluster of functionally related genes under one on-off switch. That's basically the key. Okay, and this is going to be very different than what we have. Okay, we have similar systems in place, but way more complex. But an Operon is literally saying, let's put all the genes together and all of these genes perform some sort of a function in the cell, and let's make sure we put one on-off switch in front of those genes. That's the Operon model. One on-off switch turns off the whole pathway of genes. Just that simple. Okay, so the regulatory switch, okay, or the on-off switch, is a segment of DNA called an operator. Okay, and this operator, you can see here, this is located inside of the promoter. So hopefully you remember what a promoter is when we were talking about transcription. So an operon is the entire stretch of DNA that includes the operator, right, which is part of the promoter, and then over here, the genes that they control, okay, like these structural genes in this case. And in that last picture, don't forget, these are the genes that made these enzymes so we could make tryptophan, right? Okay, so on-off switch right there, controlling all of these genes. That's the whole point of an operon. Let's see. Okay, the operon, like I said, can get switched on or off at the operator. Okay, but how do you do that? Well, it's a little protein called a repressor. Okay, and that's this guy here. Now, in this case, he's not active. Okay, inactive repressor. The repressor prevents a gene from transcribing, right? Because basically what it's going to do is it's going to sit in here and it's going to prevent RNA polymerase from jumping on the promoter and moving along making RNA, right? So how do I make this on-off switch guy? Well, here's the switch, but how do I make this repressor protein that fits inside of that switch to turn it off, for example? Well, that's a gene that's upstream in this case, and that's called a regulatory gene. Okay, these are my structural genes that are all lumped together in an operon for whatever product. Okay, and in this case, whatever process. In this case, these genes are all related to each other to make tryptophan, right? To synthesize tryptophan. Well, upstream of this operon, we have another gene that's kind of related called the regulatory gene. And the regulatory gene gets transcribed, makes RNA. The RNA goes to a ribosome to make this protein, which is going to fit into the operator to turn that gene off. That's a regulatory gene. So it's not part of the operon. So the repressor can be in the active or inactive form depending on the presence of other molecules. A corepressor is a molecule that cooperates with the repressor protein to switch an operon off, right? So for example, like we've been talking about, let's use E. coli, right? Escherichia coli. So these cells can synthesize the amino acid tryptophan, remember, as long as they're given that precursor molecule, they'll go through that metabolic pathway and make tryptophan. Okay, fair enough. So what's going on here? Well, remember we said how the smartest, maybe that's the wrong word, but the most efficient bacteria Once they have enough tryptophan, they're going to say, hey, I don't want to keep making tryptophan. That's stupid, right? Natural selection would weed out the ones who couldn't do that. Well, guess what? Here comes the negative feedback. If you make enough tryptophan inside of your cell, right, the tryptophan, which looks like this right here, that's going to go ahead and bond right here to the repressor. And look what it does when it bonds to the repressor. It activates it. So now the active repressor can fit inside of the operator and turn off the gene. Look at poor RNA polymerase. He wants to go on there. Isn't this beautiful? Think about it. You have a whole bunch of tryptophan being made, and this is the bacterial's way, the bacterial cell's way of saying, no. E. coli saying, well, okay, I have enough tryptophan right now. Why am I going to waste my time making those enzymes, those structural genes, to make more tryptophan when I'm already flush at the moment? So let's go ahead and allow this to bond to my repressor, making tryptophan a co-repressor. This will activate the repressor, which now fits into the operator, and boom, the Operon is now off. Pretty cool, right? What a neat way to save resources. Okay, so by default, the trip Operon is going to be on, right? And what does that mean? It means that the cell needs tryptophan. So of course that Operon is going to be on, so we can make the enzymes that will change that precursor molecule into tryptophan, right? However, the system works because of negative feedback. It works because as this Operon is on, you're going to continuously make tryptophan. And once your tryptophan levels get high enough, once they rise up, that tryptophan is going to start interfering. It's going to start bonding to the product of the regulatory gene called the repressor. So tryptophan acts like a co-repressor. It's going to bond, and once you bond to the co-repressor, it activates the repressor, and now you can't get RNA polymerase to jump onto this DNA, right? Part of the promoter is shut down, and now RNA polymerase can't get on there. You just stopped making tryptophan by using tryptophan, right? What a very complex and yet so simple system of saving resources and regulating your genes. Okay, so there's repressible and inducible operons, and these are both types of negative gene regulation. Okay, and these words should eventually feel very organic to you. You should just be able to talk about whether an operon is a repressible or inducible. Okay, now think about what this means. A repressible operon is one that's normally on. Oh, just like tryptophan, right? Okay, and the binding of a repressor to the operator would shut it off. So that makes a lot of sense that the trip operon would be repressible, right? Why does that make sense that the trip operon would be repressible? Well, think about it. You need tryptophan. So you want that operon to be on until you don't need it anymore, and then go ahead and turn it off. Makes sense? So what would be the opposite of a repressible operon? Would be one that is normally off until you turn it on. Now, what in the world would that be for? Oh, maybe there's something new in the environment that's not always there, and now you have to deal with it, right? That would be an inducible operon. So if you look here, an inducible operon is one that's usually off, right? But then a molecule called not a repressor, right, or a co-repressor this time. An inducer inactivates the repressor and turns on transcription. All right, let's take a look at this. The lac operon. Okay, this is one of the most studied operons, probably the most studied operon in all of biology. So the lac operon is inducible, right? And that means that it's normally off, right? The operon is off. And what that means to us, you can see here, is that the repressor protein is active. This guy is ready to block RNA polymerase normally. So this is off, okay, this operon. It contains genes that code for enzymes used in the hydrolysis of lactose. So the lac operon, the whole point of the lac operon is to make genes that helps the cell break down lactose. So, I wonder what would turn on the genes that are going to be responsible for breaking down lactose. Lactose, right? In this case, now it oscillates back and forth between lactose and allolactose, but allolactose is what we call an inducer, right? Because the gene is going to be off right? And then all of a sudden, when lactose or allolactose shows up in a system, the allolactose, as you can see here, it can bind, okay, to the repressor and inactivate it, thus turning on the operon for genes that break down lactose. And they do some other stuff. Let's take a look. Okay, so here you can see the lac operon, okay? Now, again, let's look at the bottom picture here first. You can see that there are 3 main structural genes here, okay? And you can see that these 3 genes make 3 different products, okay? LAC-Z, LAC-Y, and LAC-A. Beta-galactosidase, we know what that does. Permease, we know what that does. Transacetylase, there's still some argument going on between scientists over exactly what that does, that protein does. But basically, these genes all work together to either get lactose in your cell, okay, or hydrolyze it. Now, as you can see, this operon, it's going to be off. Poor RNA polymerase can't get on there to make the genes to break down lactose because the repressor is active. What in the world would inactivate this? Allolactose. Once allolactose comes along, it'll bond to the repressor, inactivate it, and now RNA polymerase says, oh, thank you, I can now join onto the promoter, and I can go ahead and transcribe this DNA into this RNA, and then here you can see the start and stop codons, right, for all the proteins. It can make the RNA, and that's going to make the proteins, right, at the ribosome. In this case, beta-galactosidase is basically lactase, right? This is the enzyme that's going to rip it apart, and permease helps the bacterial cell. It's a protein that helps more lactose enter the cell. Okay, very, very cool stuff. Now, one difference between this and the trip, besides the fact that this operon right here is normally off, right? So it's inducible, whereas tryptophan was repressible because it's normally on. Another difference is your regulatory gene. In the trip operon, it was well upstream. Right here, it's just barely upstream. Okay, it's actually connected to the promoter. Okay, but besides that, it's still not part of the operon, right? The operon is just the promoter, okay, which includes the operator, and then the structural genes. Okay, so let's take a look at a little video here and find out exactly how the lac operon works. Okay.
The E. coli lac operon is an example of an inducible set of genes. These genes are responsible for the breakdown of lactose into sugars used for cellular metabolism. This inducible system also involves bacterial DNA, a repressor, mRNA, and the sugar molecule lactose. This animation will only focus on 2 of the 3 proteins encoded by the lac operon: beta-galactosidase and permease. Gene expression can be induced or turned on when a specific inducer molecule appears in a cell. For inducible systems, a repressor molecule prevents gene expression by binding to the upstream controlling region. Lactose is the lac operon inducer molecule. After first appearing in the cellular environment, lactose passively enters the E. coli cell and binds to the repressor molecule. This binding releases the repressor from the controlling region. At this point, RNA polymerase can begin transcription of the operon. Here we show 2 of the 3 lac operon genes being transcribed into mRNA. Ribosomes then bind to the mRNA and the 2 proteins are translated. The first protein is beta-galactosidase, which breaks down lactose into 2 simple sugars. The second protein is permease, a membrane-bound protein. When embedded in the cell membrane, permease functions to provide a direct route for the lactose outside the cell to be imported into the cell. This import occurs at a much greater rate than the passive transfer we first observed. Because translation continues inside the cell, other permease proteins become embedded in the membrane. This further increases the rate at which lactose enters the cell. Beta-galactosidase breaks the cellular lactose into the simple sugars glucose and galactose. Once its concentration is greatly reduced, the lactose bound to the repressor are released. At this point, the repressor again binds to the controlling region and gene expression is halted. For all inducible systems like the lac operon, it is the interaction of the repressor and inducer molecules that mediate gene expression.
Okay, so pretty cool stuff, right? Some dramatic music there too. All right, so one more little thing to throw in here before we talk about positive regulation. Just remember catabolic and anabolic, right? Or catabolism and anabolism when you're talking about reactions, okay? What's happening here? Inducible enzymes usually function in catabolic pathways. Their synthesis is induced by a chemical symbol. What exactly does that mean? Think about it. If it's inducible, that means that, oh, it's probably catabolic because something new showed up and now you have to deal with that thing, probably breaking it down, right? So inducible enzymes are generally catabolic. Okay, so that's like the lac operon. Now let's look repressible enzymes. They function in anabolic pathways, right? Think about that. Think about your tryptophan, right? That's an anabolic pathway, but you want tryptophan, right? You want to anabolic reaction make tryptophan. Oh, that's why it has to be repressible. Okay, it's normally on, but if you have enough tryptophan, you turn it off. So just make sure that you can use those words pretty easily, right? Catabolic and anabolic pathways. So the regulation of the tryp and lac operons, though, are negative control of genes because, again, the operons are switched off by the active repressor. Okay? So, and when it's active, there's a lack in activity. It's not doing its job. Okay. Now, generally, that might be enough, right? We might be all finished for today and say, all right, we got it, right? Those are the two operon models we looked at, and that's how bacteria regulate their genes. There's one more step here, okay? We could also have not just negative, but positive gene regulation, okay? And remember what positive regulation is. That's like somebody putting their foot on the gas pedal, right? Negative feedback, that's like hitting the brakes. Positive feedback, whoa, that's like hitting the gas pedal and getting this reaction to happen even faster, all right? And there is a way for the lac operon to go even faster. So let's take a look here. So some operons are also subjected to positive control through a stimulatory protein such as Catabolite Activator Protein, or CAP, which is an activator of transcription. All right. So here you can see DNA, okay, in the background here, and here you can see CAP, and cyclic AMP plays a role here. Okay. Now, depending on which class you're in right now, you might not have heard about cyclic AMP, but that's a little small non-protein second messenger, we call that. Cyclic AMP is really involved in a lot of communication in your cell. Okay, it's a very, very, very useful molecule. And it basically, it's just like AMP. Okay, remember ATP, your energy molecule? It's kind of like that, only it loses two phosphates and it kind of bonds back on itself. But cyclic AMP, super, super important molecule inside of your cell and inside of bacterial cells. So let's have a look here. When glucose, which is E. coli, that's their food, right? They love glucose. Okay? When that's scarce, when there's not a whole lot of glucose around, CAP is activated by bonding with cyclic AMP. Okay? So, E. coli prefers glucose, of course, rather than lactose. So if you gave E. coli a plate of glucose and a plate of lactose right next to it, they're going right for that glucose. However, as the concentration of glucose decreases, cyclic AMP will increase. Okay. Now, cyclic AMP then bonds to CAP like this. This CAP right here is not activated. But if glucose falls and cyclic AMP rises, and then that cyclic AMP, because of the falling glucose, bonds to the inactive CAP, all of a sudden CAP gets activated. Now an active cap can bond to the promoter, and basically what it does is it increases the affinity of RNA polymerase to jump on. It's like hitting the gas pedal, right? Because we already have our allolactose around, okay? So this gene is going to be turned on, right? It's an inducible system. So the allolactose is an inducer. It basically inactivated the repressor, and RNA polymerase can now jump on. Yeah, I have lactose around. But E. coli would just be like, okay, yep, yeah, good for me. I have E. coli. Or I'm sorry, I have lactose. However, if your glucose is falling off and you have lactose, boom, cap gets activated and RNA polymerase is like, yep, let me at it. Let me make those sweet genes to break down that lactose sugar. Okay, and that's why it's positive gene regulation. And finally, when glucose levels increase again, which would make E. coli very happy, cap is going to detach, okay, and transcription will return to a normal rate. Again, if I still have lactose around, yeah, sure, RNA polymerase will jump on there because the gene is on because I have lactose around, okay? But when I don't have trouble with glucose, if glucose levels return to normal or they go up, all of a sudden I don't have my cyclic AMP anymore to activate cap. So cap basically would stay off, and this metabolic pathway just happens at a normal rate instead of at a hyper rate, okay? So basically, that's the way that bacteria regulate their genes. How do they do it? They lump everything together needed for a specific process into what we call an operon. There's one switch to that operon, right? That's called the operator. Now, sometimes the operator is blocked up with the repressor, and that would make the operon off. Sometimes the repressor is not bonded. That means the operon is on, right? In an anabolic system, the operon is usually on, right? Like tryptophan, like the trip operon, because you want to make tryptophan. So what happens is when you make enough tryptophan, tryptophan acts like a corepressor, and it comes back, joins with the repressor to activate it, and turns the operon off. Why would we make more tryptophan when we already have enough? That's an anabolic pathway. The opposite of that is an operon that is inducible, right? And that would be like the lac operon. The lac operon is generally off. Why would we keep the lac operon on unless we have lactose? But if lactose is introduced to our system, sure, lactose acts like an inducer, and it goes up to that repressor molecule and it inactivates it, right? This is an inducible operon. So what it does is it pulls the repressor off of the promoter. And now RNA polymerase can jump on there, right, to make enzymes for a catabolic reaction to break down lactose. Really cool. And we even learned a little bit about positive regulation, how if your glucose levels go down, cyclic AMP goes up, right? And that's going to keep the lac operon going at an even faster rate, because now E. coli is getting a little skittish, saying, where's my glucose? Oh, I guess I do want lactose after all, right? So bacterial gene regulation, really cool stuff. It's pretty simple once you wrap your head around it, but sometimes that takes some time, okay? And I hope this video was helpful, and good luck studying. Take care.